Hi everyone, I'm Ramin, a PhD student at UBC, and today I'll present our work at CVPR 2023. As a quick summary, in this work, we take histopathology images as input, apply cell segmentation and graph construction approaches to detect all the cells within the image and build a graph from them. You can see an example of this graph in the right figure. Then, by collecting the graphs from all the available images across different staining modalities, we can build a bundle of graphs and pass them through our network, which is called AMIGO. The outcome of this network is a prediction for the survival time of the patient, which can be used for ranking the patients and also patient stratification in clinical practice. Looking at the results, you can see that our approach can significantly outperform all of the existing baselines, including the patch-based processing ones. In the domain of digital histopathology, one of the main data points that we work with is called whole slide image, or WSI. And these images are scans of tissues that are stained with hematoxylin and eosine, and each of them can reach to something around 100,000 by 100,000 in terms of pixels. And because of this extent of the data, we cannot use the conventional machine learning and computer vision pipelines. So to deal with this extensive amount of data, the main approach and the conventional approach is using multiple instance learning, or MILs, which means we break these images into smaller pieces and then pass each of these pieces into a neural network, a pre-trained neural network, to get the embeddings, and then using different pooling mechanisms like mean pooling, variational pooling, and also attention pooling, we can aggregate these representations to come up with one single vector for the whole slide. And finally, we can use that representation for downstream tasks. But the main issue with the MIL approaches is that they are mainly focused on patches, which means they would lose information about the individual cells, like cell location, cell type, and more importantly, the mutual information and interactions that exist between the cells. On the other hand, many studies have shown that these types of information and mutual interactions can be quite important for downstream tasks, especially tasks like survival, where we need access to detailed information about the context of the tissue. So to address all of these issues, we came up with a model that we call AMIGO. In AMIGO, we take the input image, apply a pre-trained cell segmentation to find all the cells and also their cell types. Then we're going to use a graph construction approach to connect each cell to its k-nearest neighbors. You can see an example of this graph in the right figure. For each patient, we're going to have multiple images that are stained with different biomarkers, highlighting different types of cells. So we're going to apply the same procedure to collect all the graphs and create a bundle of graphs. Then we're going to pass this bundle of graphs into our network to do a prediction for the survival time of the patient, which can be treated as a score to divide the patients into high risk and low risk cohorts. In this method, we are using several novel design choices. The first one is using shared context processing in contrast to multi-modal processing. In a normal multi-modal processing approach, we usually apply a non-shared encoder to each of the modalities. But in our case, since we are dealing with graphs in all of the modalities, it is actually a better idea to use a shared unit in the earlier levels so we can take advantage of the generalization. Then in the mid layers, we can use a non-shared encoder for each of the modalities to give more flexibility for the processing. Then in the high levels, we can actually use another shared unit to unify all the representations. The shared units can be seen at these parts of the network. The second design choice is using a sparse processing. More specifically, we apply a masking operation over the nodes to reduce the number of nodes and also edges in the graph for more efficient processing. Even though in the first glance, it seems like the model won't be able to propagate the message between the nodes, surprisingly, we realize that the network is so robust that it can still keep the same performance with higher levels of sparsity. And our last design choice is using a batching mechanism to make sure that the number of patients that have survived is constant throughout all the batches within the training. And the main reason for this is stabilizing the gradient flow within our training. For more details on this, please refer to the paper. In this study, we have used two different datasets. The first one is INEUT, which is a high-grade serous ovarian cancer dataset, which is the most aggressive subtype of ovarian cancer. This dataset includes 1,600 images across 188 patients, which is going to be released with this paper for more research. MIBC is the second dataset that we used, and it's from muscle-invasive bladder cancer dataset, which contains 585 images across 58 patients. As for the evaluation metrics, we rely on two most commonly used ones. The first one is C-index, which measures the agreement between the predicted and actual survival times for the patients, and it ranges from 0 to 1, with higher values showing more agreement. The second metric is p-value, which measures how well the actual survival curves of high-risk and low-risk cohorts of patients are different. When compared to the baselines, our approach can actually significantly outperform all of them, including hierarchical vision transformer. And it can do so across both datasets with less parameters. This is actually quite interesting because our approach is the only one that can achieve a significant p-value in the INEUT dataset. And this is the first time that a study has shown the separation in the high-risk and low-risk cohorts of high-grade serous ovarian cancers with only imaging data. To be more specific about the separation of the cohorts, we take the median of the predicted survival time within each dataset and then divide the patients into high-risk and low-risk cohorts based on the comparison of the predicted survival time with this median. Then we take a look at the actual survival curves of these two cohorts, and as you can see, in both datasets, there is a clear separation between the actual survival time for the high-risk and low-risk cohorts. To make sure that our design choices are reasonable, we have performed extensive ablation studies on different parts of the network, and these experiments show that our design choices are actually important for the performance of the model. Specifically, as for the sparsity, as we increase the ratio, we can see a drop in the training flops, which is expected. But in contrast, as for the C-index, we can see it's almost constant with different sparsity ratios, which means that our model is robust enough to propagate the information between the nodes even with high degree of sparsity. Also, as for the BCP or batch sensor portion effect, you can see that a specific ratio of the BCP can enhance the performance of the model because of the gradient flow. So in conclusion, we developed a biologically inspired and cellular-based approach that can outperform state-of-the-art methods in survival prediction. We also introduced several novel techniques such as shared context processing and batch sensor portion, which can be used in further research. Also, for the first time, we showed that the separation of patients in high-grade serous ovarian cancer can actually be done by using histopathology images only. And finally, we released a large dataset of immunohistochemistry images with survival information, making it one of the largest datasets in this context.